Welcome to the first episode of Subject Forum in Biology. Today with me we have Mrs. Aishat Wahida, the leading teacher in the science department from Hiria School, very experienced biology teacher. She joined in the teaching field since 1999. And also we have Mrs. Fatma Sasla from Tajuddin School. Welcome both of you to this show. And in this forum we will be clearing your doubts in various topics, especially in inheritance and protein synthesis. And we would also like to take the questions that you have asked from various other topics. To begin with, we have some questions from inheritance, like what is inheritance, the difference between phenotype and genotype, difference between gene and allele, difference between complete dominance, codominance. So various questions are being asked from this particular topic, and Mrs. Fatma Sasla will answer them.
All right, to start with, let's focus on what is inheritance first. Before I explain what is inheritance, let's know what is genetics. Genetics is actually the branch of biology which deals with inheritance. It is the study of heredity and variation among species. Now, here the heredity, that is inheritance. Inheritance is actually the transmission of genetic materials from one generation to the other generation. Basically, it is from your parents to you, from parents to children. That is inheritance. And then the other parts. The most important thing is inheritance here is genes, and the genes are actually it is passed on from parents to the children. Here, first of all, let's see how these genes are passed on from parents to children. Okay, to start with, many students they actually have the problem in identifying what is the difference between a gene and allele, and what are chromosome, and what is the role of chromosome in this inheritance. First of all, let's take a cell, and students, you must know where in our body, where in the cell the chromosomes are found. Let's take a cell, and inside the cell the chromosomes are found in the nucleus of the cell. In the nucleus of the cell the chromosomes are found, and these chromosomes are actually, it acts as the vehicles to transport these genetic materials from the parents to their children. So let's take this, and it is this in the form of chromosome, it is transferred to the children. Let's say this is the parent cell, and this is the offspring. The chromosomes, the chromosomes are passed from parents to the offspring. Let's recall something that you have already learned, that is fertilization. At the time of fertilization, the offspring receives two sets of chromosomes. That's one set from the mother and one set from the father. That is actually how an offspring receives the chromosomes. And these chromosomes, the one that, let's just say this is the parent, mother, and this is the father. I'm sorry, this one is a little bit small. And at the time of fertilization, the offspring receives one set of chromosomes from the mother and one set of the chromosomes from the father. Altogether, we humans, we have 46 chromosomes. So it is 23 from the mother and 23 from the father. So the genetic material in the parents are transferred to the offsprings in the form of chromosomes. And these chromosomes Actually, let's look at the detailed drawing of the chromosome. The chromosome here, each chromosome is actually a coiled DNA molecule. It is a coiled DNA molecule. And what is gene here? Gene is a section of DNA which is actually coding for specific trait or specific protein. Actually, when you write the definition for gene, you must write it is the section of DNA which codes for production of a protein molecule. That we have to say that we have different traits. Our eye color is due to a different protein molecule. Our hair color is due to a different protein molecule, and our hair type is also due to another different protein molecule. That's why actually when we define gene, we have to define it as it is a section of DNA. For example, this section of DNA this could be the one which is coding for eye color. This could be the one which is coding for eye color, and if you take this section of the DNA, it could be the section of DNA which is coding for hair type. All these are traits, in other words, characteristics. Each and every trait that we have in our body, it's due to genes, and these genes are found on chromosomes. Chromosome, it will have lots and lots of genes in it, and the gene, when we actually, this is the chromosome, how it will be when we look at it, and then if we go into details of it, it is actually a coiled DNA molecule, and the section of the DNA molecule, the length of the DNA molecule which is coding for specific protein production, that is what we call as gene. Alright students, that's about genes, and then from this topic, you after learning this topic, you will actually it will give you answers like why you have brown eye color while your sister is having black eye color, and why you are having curly hair while she is having straight hair. All these variations through this topic, actually you will get to know answers of these questions. It is really an interesting topic when you go into details of these things. Okay, then I'll move on to explaining what is actually the difference between a gene and allele. So far, I hope you have understood what is a gene. What do we mean by a gene? And a chromosome, when we take a chromosome, it is full of genes in it. So the offspring will be inheriting one set of chromosome from the mother and the other set of chromosome from the father. So you are a combination of your mother and father, obviously. All right, students. Now, gene. Let's have a look at this diagram. Okay, you know that now we have 46 chromosomes, and out of that 23 you have received from your mother and another 23 is from your father. And when these, at the time of fertilization, when these two set of chromosomes, when they combine at the time of fertilization, when a female gamete and male gamete fuses together, giving rise to zygote, that is the moment where all the things about you will be decided, your eye color, hair type, all the things will be decided at that moment, your intelligence even. Okay, so at that moment when these two sets of chromosomes, when these align, it will be aligning in such a way that homologous chromosomes will pair up. Homologous means it should be similar in shape, similar in length. These homologous chromosomes pair up, and let's say this is the one you have received from your father and this is the one that you have received from your mother. Here I am showing a homologous chromosome. And on this one, let's say this is a section of DNA which is coded for eye color. Eye color. Here this is the gene. Eye color is the gene. And maybe your father is having black eye color. And the one, your mother may be alleles. Gene is a section of DNA that is coding for specific protein molecule. Alleles are different forms of the gene. For example, here the gene is eye color. Black, blue, hazel and other colors, those are alleles, different form of this gene eye color. eye color is the gene, and the alleles are blue, black, and the other colors, other eye colors. Your mother may be having blue eye color. So here gene is gene, eye color, and alleles are the blue and black. Alternative forms of this gene, alternative forms. So from this diagram I am showing two alleles.
Can I interrupt? Eye color is a characteristic feature, you know, and then those are the different ways how it is being expressed out.
Exactly, sir.
Okay.
Yeah. So here,
I mean, you say gene is the length of DNA that controls that character, and alleles are
different
the alternate forms of the
different
gene.
forms of the exactly. So the different forms of the these eye color is the gene, and the black and blue are the different forms of it. That's the alternative forms of this gene. So they become the alleles. And so for here, these are alleles, and another diagram. Let's say here these are A and B. I'll label this as A and B. A and B are alleles, but here A and C. Look at these two, A and C. These are not alleles, but different genes. These are two different genes, not alleles. A and C, not alleles, but genes, but different genes, because if it is an allele, it should occupy the same locus or same space, same location of the homologous chromosome. Here it is occupying the same location. So this becomes the allele. These two are A and B are alleles, while A and C are not alleles because they are not occupying the same location of the homologous chromosome. They are appearing on different locations. So these two are two different genes, but not alleles. Most of the students find this part difficult to identify alleles and genes. If you get a diagram like this, A and B are alleles because these are the A and B are alternative forms of the gene eye color. Eye color is the gene here because gene is a section of DNA that is coded for production of a protein molecule. Here we are talking about eye color, and A and B it becomes alleles because these are two alternative forms of the gene eye color, blue and black. And then here these two are different genes, not alleles, because they appear on different location of the homologous chromosome. This could be, let's say this is eye color, so this could be your hair type, gene that codes for hair type. So these are two different genes, not alleles. All right, students? Okay, then now after...
And again, you are also supposed to know that there will be 46 chromosomes in all the somatic cells in humans. That means all these genes will be there in all the cells, but the specific gene will be expressed out only in a specific cell. So the genes will be there in all the cells.
For example, if it's an enzyme production, so that particular gene will be expressed only at the time of enzyme production because it is not exhibiting at all time. They are very specific.
For example, we have, if we take our body, we have somatic cells and gamete cells. Somatic cells are all the body cells, and each and every somatic cell will have 46 chromosomes. So because they are diploid cells. But gamete cells, they are haploid cells. Haploid means they have half the chromosome number of the somatic cells. Somatic cells, if you take human, for example, we have 46 chromosomes, and the gamete cells, the gametes that human produce will have 23 chromosomes. These 23 chromosomes, we call it as haploid cells because they have half the chromosome number of the somatic cells. And we have in each and every...
It's always better to define diploid and haploid as diploid cells having having two sets of chromosomes, and haploid cells having one set of chromosomes, instead of stating half and full set.
And having the same, having said that the same number, the first 22 pairs of chromosomes are the autosomes, which is the body cells, and the last pair of chromosomes, even in a chart if they see it, the last pair represents the sex chromosomes. Either it could be XX if it is identical, or if it is X2 different, it will be, right?
Yes.
So out of this, the first 22 pairs are diploid cells, and the last bit will become the haploid cells in the end.
Yeah, that is what we want to add here is that when we have body cells, in our body cells we have these 46 chromosomes, that is 23 pairs. All those up to 20, the last pair, that is the 23rd, that pair is actually the pair of the chromosome which decides the gender of the child. If it is XX, when we look at the genetic diagram, when we look at all these alleles in it, it will be similar in size if it is 2X, and then we can say it is a female, it is a girl. And if it is XY, which is of two different, which will be of two different lengths, X will be longer and Y will be shorter, and that is a boy, male. Okay, and that's all about chromosomes. And then let's move on to genetic diagram, where we're explaining the phenotype, genotype, and those terms. Okay, here first of all, let's take an example of when we call some a feature, a trait is dominant, and when we call it is recessive. Okay, first of all, let's take two flowers, two plants with two different colored flowers. One red flower and the other one is white flower. Okay, the red flower one here, this color, first of all, phenotype, it is the physical appearance. Phenotype, it is the physical appearance of that organism, what we can see outside, that is the phenotype. Here, if you look at this, the red is the phenotype, white is the phenotype. And when we cross this, the genotype is the genetic makeup of it. Okay, the genetic makeup of it, it is the that is the genotype. Okay, in general, first of all, I will do a very simple one, and then I will go into detailed genetic diagram. This is just to explain the dominant and recessive trait. When we cross a red flower plant with a white flower plant, if F1 generation, if all the offsprings are having red colored flower, this means the red is dominant over white. When we cross a red flower plant with a white flower plant, if all the resulting plants are having red flower, then it means red is the dominant, red is dominant over white. So this is the time we call it as a dominant trait. This is the dominant trait, and that is the recessive trait here. Red dominant and white is the recessive. Okay, with this we are going to explain about dominant allele and recessive allele. Now, when we do genetic diagram, we actually use this phenotype, genotype, heterozygous, homozygous, recessive, homozygous, dominant, all these terms. With this, I am going to introduce you all these terms. First of all, red and white. Now that we know red is dominant, let's take a letter. We use a letter to draw a genetic diagram. Let's take a letter for this. The rule is for the dominant trait, for the dominant character, we should use a capital letter. Let's take, let's use a key, and then let's take red, capital R as red, and white as simple R. We can't take W here because this is the rule of monohybrid inheritance. We have to use the same letter and its uppercase letter and lowercase letter. Here, if it is dominant, we can use the capital letter. If it is recessive, we have to use the same letter but its simple form. So here, red, capital R, and white, that is the simple R. Here, this R, it is representing the dominant allele, and the white is representing, the simple R is representing the recessive allele. When two contrasting traits are crossed, the one that gets expressed in the first generation, that's the one we call it as dominant trait. and the one that gets suppressed here, red is expressed in all the offspring. So it becomes the dominant trait, and white got suppressed. So that is the recessive trait. And the dominant trait here is because of dominant allele, and the recessive trait is here because of recessive allele. So now the dominant allele here is capital R, and the recessive allele is simple r. Let's draw a genetic diagram here. A genetic diagram, first of all, I will go for a genetic diagram first. In a genetic diagram, first of all, you must write parent, and then parent phenotype. Phenotype is the physical appearance, red, white. Then genotype.
So you can just tell the difference between the terms so that we can
genotype,
go on.
Here the genotype, okay,
Yeah,
first of all, the genotype, let's say if it is a red color one, there are two options, right? Because red is the dominant one. To save time, I will not go into details of the genetic diagram. I will just go through the terms. Because red is dominant, so it has it can be expressed in two ways. One way is homozygous dominant. Homozygous dominant means getting two dominant alleles. Its genotype, these are the terms we use to actually explain the genotype of organisms. Okay, homozygous dominant, heterozygous. Heterozygous, hetero, the term itself tells you it's different, two different things. Hetero means one capital R, one simple r. It means one dominant allele, one recessive allele. Then it becomes heterozygous. Many students tend to write heterozygous dominant. You don't write that in genotype, but heterozygous, simply you can write heterozygous. It is their offspring, if their offspring is a heterozygous for genotype, should have one dominant allele and one recessive allele. Then the other one is homozygous recessive. Homozygous recessive means from the letter recessive, homozygous means same. So if an organism's genotype is homozygous recessive, it should have two recessive alleles. And I want to highlight this thing here, homozygous dominant, and a dominant trait can be expressed even if it is in homozygous dominant state or even if it is heterozygous state, because it is capable of expressing it even in the presence of one allele. The dominant trait can be expressed even if there is one allele of it. But homozygous recessive, the recessive trait can be only expressed if the organism is having two recessive alleles. For example, if white is recessive, if an organism to have white colored, if the plant is to have white colored flowers, it should receive two simple alleles. Here, homozygous dominant, capital R, heterozygous, it is one dominant allele, one recessive allele. Homozygous recessive, it is two recessive alleles. So in this condition, it will be white. Here it will be red, and other one it will be red, because it is able to express its phenotype even in the presence of one dominant allele. So this is about the phenotype and genotype. Phenotype, once again I will repeat, phenotype is the physical appearance of the organism. It can be the color of it. For example, in this example, a phenotype is red, white, and the genotypes, we use three different terms to identify an organism's genotype, because by looking at an organism you won't know whether that organism is having a homozygous dominant or heterozygous genotypes, specifically because a dominant trait is able to be expressed in these two ways also. But if it is a recessive trait, it will be very easy. It should be homozygous recessive for that organism to express a recessive trait. Okay, I hope you have understood these terms. Then I'll add more in my later explanations.
Thank you, Sasla Miss, for that. Now we have a few questions on protein synthesis, and there are a lot of students asking about how the protein synthesis is being carried out, the difference between the base sequence and the amino acid sequence that takes place, then where and how the proteins are made, then how the genetic code works, how the triplet codons, how do they code for specific amino acids during protein synthesis, the role of ribosomes, the role of different types of RNAs, and over to Mrs. Aishat Wahida to answer these questions.
Okay, thank you, and let's start. Like, why are proteins so important for us? Protein is the base of our life, and synthesis means manufacture. So where are proteins manufactured? That is the first question. Proteins are manufactured in the ribosomes. Ribosomes are cell organelles which is actually produced by the nucleolus of the nuclei, and then it is being sent into the cytoplasm of the cell. And ribosomes are called as manufacturers or the factory where the proteins are produced. And how do we produce these proteins? So first of all, there are three types of genetic materials. One is the DNA material, which is mainly found in the nucleus, as I was talking about DNA. DNA is a protein molecule which is found in the nucleus of the cell. And how are these DNA copied and transferred? So for that we need RNA materials. What is the difference between a DNA material and an RNA material? DNA means deoxyribonucleic acid, which is double helix coiled, and RNA is a single strand, and it is on the ribonucleic acid. And RNAs could be mRNA, tRNA, and even RNA. So first we start with the nucleus. So in the nucleus there is a DNA material. The DNA material does not come out of the nucleus at all. It remains in the nucleus. But with the help of some enzymes, they will unwind. So once they unwind, a part of this DNA material has been copied down with the help of a RNA material, which we call as the messenger RNA. And yesterday's presentation, it was shown very well by Mr. Anthony that explained this very well, but I will be adding on to that a little bit. So it is the messenger RNA which is produced in the first copy of it, that is with the help of the RNA. And in this, the RNA materials, they have these codes. So these are nucleotides. They have said what are the bases, what are the proteins, amino acids. So these are the nucleotides which makes up a codon. Three of these bases, nitrogen bases, are called as a codon. For example, ATG, which is a codon which codes for one particular amino acid. Likewise, we have GCT. But during this process of manufacturing mRNA, they will be unwinding and taking that segment. But in this, there is no thymine. So thymine will be always transferred with the help of uracil. In this base pair, in the 4 nitrogen bases, adenine will pair with thymine and cytosine will pair with guanine. But in this case, when it is a messenger RNA comes in here, we do not have thymine. So adenine will pair with uracil. And like this, they form the molecule. So once they form, there is a start signal. And once the protein is manufactured here, then they have a stop signal. Now this is removed from the nucleus and moves out into the cytoplasm. So once it moves out there, the messenger RNA moves into the ribosome. Ribosome are the organelles which is found in the cytoplasm of the cell. And inside this ribosome, protein synthesis takes place. So where is protein synthesis taking place? Your question? It takes place in the ribosome. And once it reaches the ribosome, there is another RNA which we call as the transfer RNA. So while on its way in the cytoplasm, there will be a lot of nucleotides. So each transfer RNA will be picking up these nucleotides, which is called as a codon. That codon which comes and fits exactly into its place. These have particular shapes, like each one will fit into only exact. It's like just the lock and key hypothesis of enzymes. The key can fit only to that particular lock. So likewise even here, this can come and fit into this. So once it fits into this codon, this transfer RNA is now set free to go and collect another set of. So here likewise there will be. So once the 2 amino acids are formed, 3 of these bases together we call it as one amino acid. That is a codon. So like that once it's 2 of these amino acids are formed, they start forming the peptide bonds. And how long it takes for it to form, it depends on the size of the protein molecule. It depends on the size of the protein molecule. Like according to the theory, it takes about 6 seconds to form one amino acid. I have gone through the research, and then it says, so it all depends on the type of the molecule which we are going to form. So once this is copied down, now it is the protein is manufactured in the ribosome, and this protein is ready to leave to perform a particular function. So likewise, all these proteins are synthesized in the ribosome.
I think we will have a break now, and we'll continue with the show after a short break. Welcome back to the show of Subject Forum on Biology. And to continue, we have some more questions which are being asked by the students. Many of the students, they have doubts on menstrual cycle, especially the role of hormones in menstrual cycle. The Miss will explain about that.
Menstrual cycle is controlled by 4 hormones. That is the FSH, which is the follicle stimulating hormones. As we all know, girls are born with number of ovaries in their egg cells, but it is not mature only on the onset of puberty. This is mature. So once the egg cell starts maturing, that is with the help of the follicle stimulating hormone, which is produced by the pituitary gland. And once the follicle stimulating hormone, it develops the follicle. In the meantime, they are also producing estrogen. Then once these two are produced, there is a hormone from the luteinizing hormone from a pituitary gland of the brain, which releases the egg cell into the oviduct. And once it is released, now the lining is thick and ready for fertilization. So the progesterone, which is the empty follicle, will produce as progesterone, and progesterone will maintain till birth. If fertilization does not take place, this egg will disintegrate, integrate, and then it will be coming down. So basically, the hormones are FSH, estrogen, luteinizing hormone, and the progesterone.
Then some students, they have asked questions on eye, especially regarding pupil reflex. Sasla, Miss, can you explain them?
Now, pupil reflex, there are 2 things, that what happens in dim light and what happens in bright light. When the person goes into bright light, what happens is the pupil gets smaller. Pupil gets smaller because it is controlled by a pair of muscles, which is that is the radial iris muscle and the circular iris muscle. Students, you must know that these 2 are the muscles which controls the pupil reflex, the circular iris muscle and the radial iris muscle. In bright light, what happens is the circular iris muscle contracts and the radial iris muscle relaxes. So the circumference of the pupil decreases, the size of the pupil decreases, and this is actually there to prevent any damage to our eye due to bright light. So due to to prevent any damage to the retina because of the bright light. Then the other side is what happens in the dim light. Dim light, exactly the reverse happens. What happens in dim light is, that the circular iris muscle will relax and the radial iris muscle that contracts. So it is actually increasing the size of the pupil, allowing more light to enter so the person can see clearly. That's the simple thing. Students should keep these 2 muscles in their mind, and they should know these 2 are actually working antagonistically. Antagonistically means opposite to each other. These 2 they should keep in mind, and then we can answer the questions which are on pupil reflex.
Since you have started with the eye, some of the students, they have doubts in accommodation of the eye also.
All right, accommodation is actually the ability of the lens to change its shape according to the distance the person is viewing. For example, if you are looking at a faraway object, something happens to increase the focal length of the lens so that we can see the object clearly. Okay, I'll explain what happens when we look at a faraway object. First of all, you should know here in accommodation, the suspensory ligaments and ciliary muscles and the lens. These three things are working: ciliary muscles, suspensory ligaments and the lens. Here what happens is when you look at the faraway object, the suspensory ligaments are pulled tight. Suspensory ligaments are actually, you can think it of as strings which are holding lens from both the sides as well. So when you look at in a faraway object, what happens is these two suspensory ligaments are pulled tight. So this actually exerts a force on the lens, and then the lens is pulled thinner and longer, which is increasing its focal length. So the light rays from the distant objects can actually focus on the retina exactly, forming a clear image. And then when we look at a near object, for example, when you read in a book, what happens is exactly the opposite happens. The suspensory ligaments are slack, and that's the term that you use. You don't use the relax. It becomes slack, and that is that this is actually pulled tight and slack, and you can think of it like this. When it is slack, and what happens is the force that was exerted on the lens, it decreases, and the lens becomes shorter and thicker, which decreases the focal length of it. So the light rays from the near objects are focused onto the retina, and you are able to see a clear image.
And in the examination, students, they get confused with what kind of muscles they have to mention in the answer. And always remember, when they are talking about pupil reflex, it is the iris muscles they have to mention. And when they talk about the accommodation, it should be the ciliary muscles.
Students often tend to do that mistake. Yeah. That is a good point. So you highlighted that. So they always somehow they mix these two and then they write it. But in focusing on the pupil reflex, it should be iris, because iris is the one which is actually controlling the amount of light that enters into our eye. And in accommodation, they should be focusing on the ciliary muscles. And the more specifically, circular ciliary muscles will do. No need to focus more on the radial ciliary muscle, circular ciliary muscle, and the suspensory ligament, and the changes that are brought to the lens.
Now coming to an ecology question, some students, they have doubts on eutrophication.
Eutrophication, the term itself implies the excessive growth of algae. And there should be reasons why this algae is growing. If you look at a train traveling from a distance, when you see the ponds, it's green in color. But when we go very close to it, and if we take water, it is not green. Water is colorless. But that is a reflection of the algae which is growing there. So when they leach nitrates or phosphates, or even from home the detergents, these are nutrients where it absorbed by the plant by active transport or the algae. And then excessive growth of algae, we call it as eutrophication. And consequences of eutrophication: once the whole pond or the water body is filled with algae, it forms a carpet around. So there is less light penetrating right into the deep into the waters. But there are organisms which is living inside the waters. So these organisms will not get enough oxygen and light. So therefore, finally, these organisms will die. And once it dies, the algae itself will start dying, and the bacteria will be using up all the oxygen. And that condition we are calling as biological oxygen demand, where all the oxygen is being used up by the microbes, so all the organisms die. So in other words, eutrophication is an excessive growth of algae due to leaching of nitrates and phosphates from households or factories or any form.
Some students, they have doubts on double circulation and the two circuits which are involved.
Now, double circulation, actually we call it as double circulation because in one complete circuit of our body, the blood passes through the heart twice. That's why you call it as double circulation. And in double circulation, there are two parts you have to talk about, students. The first is pulmonary circulation, and the other one is systemic circulation. Pulmonary circulation, it is the circulation of blood between the heart and the lungs. Here, you can talk about pulmonary circulation starting from that is the point where when the deoxygenated blood, students, you should know to explain this, you should know the structure of the heart very well. There are 4 chambers, and the upper 2 chambers are atria, then the lower 2 chambers are ventricles. Now, the right side is actually the side which it pumps the deoxygenated blood, and the left side is the side which is pumping the oxygenated blood. Here, once when the vena cava fills the deoxygenated blood in the left, in the right atrium, then it goes into the right ventricle through the tricuspid valve, and then from the right ventricle it will be pumped to the lungs through the pulmonary artery. And the pulmonary artery, it carries this blood, deoxygenated blood, to the lungs, and then there it gets oxygenated, and then the oxygenated blood is brought back to the heart through pulmonary veins. Students often tend to get confused whether pulmonary artery is carrying oxygenated blood or deoxygenated blood, because most of the arteries will be carrying oxygenated blood. But students, you can keep one thing in your mind: if it is an artery, it will be always carrying blood away from the heart. That artery, the A stands, from that you can keep that in your mind. Arteries always carry blood away from the heart. And then once that is the pulmonary circulation, that the circulation of blood between the heart and the lungs. And in the pulmonary circulation, the pressure will be lower than that of the systemic circulation. In the systemic circulation, what happens is the oxygenated blood is pumped from the right ventricle of the left ventricle of the heart, which is more muscular. That is why it generates more pressure when it contracts, and then it pumps blood to the aorta, which carries blood to aorta is a main blood vessel, main artery that carries oxygenated blood, and then aorta pumps blood to all the parts of the body, the oxygenated blood. And systemic circulation, the pressure will be higher than that of the pulmonary circulation, because it is the systemic circulation is more extensive than the pulmonary circulation, because in systemic circulation it has to pump blood to all the parts of the body.
We are talking about double circulation basically because the blood passes to the heart twice before it reaches to the rest of the body. So in short, that is what we are talking about, and double circulation.
The pressure is high in the systemic circuit. That will also help in bringing about the ultrafiltration of blood in the kidneys.
The important substances such as glucose, oxygen, and these things.
Then, Wahida Miss, can you differentiate between nitrogen fixing and nitrification?
Nitrogen fixing, that is fixing the atmospheric nitrogen to there. So as we know, we have 78% of nitrogen in the atmosphere, but which cannot be used directly as carbon dioxide and oxygen. Carbon dioxide used by plants and oxygen by us, but this nitrogen cannot be used as it is. So the most usable form of nitrogen in this earth is nitrates. So nitrogen is converted into nitrates by 2 forms. One is by the fixation, that is the biological fixation, which there are bacteria which is living in the root nodules of leguminous plants. They convert this atmospheric nitrogen to nitrates. And during thundering and lightning, because of the high energy liberated, they combine with oxides of nitrogen, forming nitrates which falls onto the earth, and that is also converted back into the nitrates. So these are ways which we call this nitrogen fixation. They are fixing the atmospheric nitrogen. Whereas nitrification is where there is a group of bacteria which lives in the soil, where first of all when we die it undergoes decomposition, and decomposition releases urea and many other ammonium compounds into the soil. So these ammonium compounds are converted into nitrate. Now first of it is nitrites, later oxidized to nitrates, and nitrates can be absorbed. So nitrification is a process where ammonium compounds are converted into nitrites and nitrates, whereas nitrogen fixation can be directly the atmospheric nitrogen into nitrates with the help of microbes.
And also it can be the formation of ammonium ions, especially in the leguminous plants, in the root nodules. Okay.
I always explain it to my students. There is an easy way to understand this. If it is nitrogen fixing, always it should start from nitrogen gas. That should be the starting point, should be nitrogen gas.
The nitrogen gas later it's converted back into its nitric acid and other forms, so they should know the differences.
Then another question from the circulatory system, the importance of platelets and also the importance of white blood cells.
Okay, so, but the platelets, some students tend to actually think they are blood cells. Students, it's not blood. It's not a blood cell actually. Platelets, it is very important in our body. Actually, it is having its function is blood clotting. So that's very important. That it helps in blood clotting. When there is a cut in our body, it helps to clot the blood. Okay. So that is very important, the role of platelet in that. Then the other one is white blood cell, namely that you have to know about two white blood cells, that's phagocytes and lymphocytes. Phagocytes, white blood cells as a whole, it has a function of body's defense mechanism. It is actually protecting the body from any foreign object, any pathogen that is entering our body. In that, the lymphocyte, what it does is the lymphocyte will be producing antibodies. These antibodies actually neutralizes the toxins released by the foreign body which has entered into our body. It could be a pathogen which can be cured without any antibiotics taken, without even if you don't take antibiotics. Then the other one is, so lymphocyte, it produces antibodies, and then the other one is phagocyte, which carry out phagocytosis. Phagocytosis, it is engulfing when there is a foreign object in foreign body, like pathogen or any other thing which has entered in our body. This phagocytes, it actually engulf it, and it inside the phagocyte, it releases enzymes and digest and destroy it. So white blood cell altogether, it has a defense mechanism in the body, phagocytes carrying out phagocytosis, and the lymphocytes, it will be producing antibodies.
And always remember that platelets, the main role of these platelets is to release the enzyme known as thrombokinase, and white blood cells, they have a major role to play in providing the immunity to the body. Coming to immunity, Wahida Miss, can you explain about passive and active immunity?
Thank you. And when Shazla was talking about antibodies, the function of antibodies is not only to deactivate enzymes, but they also helps to clump all the microbes, so it is easy for the phagocytes. This goes hand in together. Yeah, right. So immunity is, we are talking about our immune system, which again we are talking about white blood cells. So we have two types of immunity, which is in the new syllabus IGCSE we have this topic. So it is active immunity and passive immunity. Active immunity is by either you get the disease or by vaccination. What is a vaccine then? A vaccine is a weakened pathogen which is entered into our body. So the minute the pathogen enters our body, lymphocytes will recognize, and then they will start producing antibodies. These antibodies are proteins. Earlier to that we discussed the importance of proteins. So these proteins will have a particular shape which can fit into the antigen which is around the microbes. So then they will be able to destroy it. And as we go on in our body, then naturally lymphocytes will start multiplying, and all the lymphocytes are not producing antibodies. Some of these lymphocytes remain in our body as memory cells. So they remain dormant. Some of it is producing. So these memory cells remain in our body. So next time when this microbe, let it be a virus or bacteria which enters into our body, so these memory cells will recognize very easily, very quickly, and then they will be able to produce the antibodies. So before the person falls sick next time, person will be able to recover. But today in this world, right now we are going through with coronavirus because we still don't have a vaccine, and we do not have produced any antibodies to fight against it. So it is taking for the time for the person naturally to develop these antibodies. That is why people are getting cured. But in the meantime, people are dying out because it is taking a long time for these antibodies to develop. There are no memory cells because it has not entered before. So then that is what we call it as the active immunity. Then we have the passive immunity, where in the passive immunity, the antibodies which has been produced outside is injected, that is for short-term goals. So if an epidemic breaks out and if there is an antibodies which is already produced, so we can take it. And these will not last in our body for a long time. This is only for temporary. And then after the disease it's over. But it is best to have an active immunity, so we will be able to fight, defend against any of the microbes which enters into our body.
I would like to highlight one thing. Some students actually when it comes to vaccine, they should specifically know that it is for a disease which is caused by a virus only, because virus cannot be killed or destroyed by antibiotics. Antibiotics can be given only for a bacterial disease, anything which is caused by bacteria. So vaccine is therefore something which is caused by virus. And about the coronavirus, yes, that COVID-19, that coronavirus, we haven't still found any cure for that, any vaccine for that.
Antibiotics are effective against fungi and bacterial diseases. That is why we are talking about vaccines.
That's why they are
vaccines are weakened pathogens which is entering into our body.
Weakened in virus specifically, right?
Let us hope the world will find a vaccine for coronavirus.
Yeah.
Definitely.
That's
if not, antibodies produced for it.
And coming to synapse, and the transmission of impulses takes place across the synapse by means of the neurotransmitters. And they are asking, how does the neurotransmitters work?
Actually, students must know when there is a neuron, that nerve impulses are carried along the neuron, and then we have that arc is that there are 3 neurons actually. When for some coordinated behavior to occur, an impulse must pass through these 3 neurons, namely sensory neuron, relay neuron, and motor neuron. And between these 3 neurons, at the junction of each neuron, there is this synapse. So when for example, if a nerve impulse is carried along sensory neuron, and then when it reaches its end plate, there are something structure known as synaptic knobs. Then this synaptic knobs, actually it has something known as synaptic vesicles. These vesicles, it releases neurotransmitters, and then between 2 neurons there will be an empty space which is known as synapse. That is the gap between these 2 neurons. So these neurotransmitters will be released to that gap, and then it diffuses across that synapse, and then it diffuses and then binds to the receptors on the next neuron. That's basically how a nerve impulse is passed on from one neuron to the other across a synapse.
And neurotransmitters are chemicals.
These chemicals will diffuse across this gap,
Yes.
and these chemicals are being accepted by some receptor proteins on the other side, and they are complementary in their shape, so they get attached, and as a result of that, a new electrical impulse will be initiated at the other end of the neuron.
That is when all these proteins, they have a particular shape compared to carbohydrates proteins. So that is why we say proteins have a high attitude, because it is amino acids, right? Yes. Because they have a particular complementary shape to intuit.
Different traits. Each trait is because of a different protein molecule, right? Different types of protein.
Coming to genetics again, they want to know what is mutation, and specifically what is gene mutation.
Okay. Mutation is a sudden change in the chromosome. It can be either on the genes or it can be on the chromosome number. So it's just a sudden change, and these normally these mutations are caused by the exposure to radiations we have seen and gone through during the First World War. Hiroshima and Nagasaki, they have atom bombs. So there are a lot of mutants genes moving around even in Japan. Even till now, this can be passed on from one generation to another generation if it occurs in the sex chromosomes. Otherwise, it is limited to the person. So mutation is a sudden change in the chromosome number or in the genetic makeup. So gene mutation refers to the change in the gene. For example, we are talking about sickle cell anemia, where we are forming a defective hemoglobin. The hemoglobin produced, the hemoglobin is also a gene which is produced. So here, if one of these nucleotides is being replaced by another, so therefore it forms a nonsense gene or defective gene.
Okay, thank you. We have answered some questions. There are plenty of other questions remaining in our list, but we have the time constraints. So hope we would be able to answer all your questions in the coming episode. Thank you for joining us, both of you, and thank you for watching.